Hello everyone. In this video we are going to discuss about the basics of biomedical instrumentation. So in this we are going to discuss about the bioelectrodes. Bioelectrodes is nothing but an interface which acts between the human body as well as the electronic system. So the ionic current which has been produced inside the body is going to get converted into the electronic current which is easily acceptable by the electronic device as well as which is used to going to measure the biological signals such as ECG, EMG and EHG. So there are three different types of electrodes: body or surface electrodes, which is used to measure the biopotentials from the surface of the body; microelectrodes, which is used to measure the potential within a single cell; whereas the depth and needle electrodes, it is used to measure the electrodes which could be deeply penetrated inside the skin. So in this video, let's now discuss about the second type of electrode, which is the microelectrode, which is used to measure the potential within a single cell. So there are two types of microelectrodes. One is metallic electrode and the other one is non-metallic electrode, and both are used to measure the electrodes within a single cell. So it is used to measure the electrodes or the biopotentials within a cell. It is also called as intracellular electrodes. So let's see the both the types in detail now. First is metal electrode or the metallic electrode. So it has a metal rod. The metal rod has been insulated by an insulating material, and the tip of the metal rod has been inserted into a single cell where the cell has a cell membrane as well as the nucleus. So this is what about the metal electrode. So always a metal electrode has to be have a reference potential. The single metal rod whose diameter is very small, it has been penetrated inside the single cell, and it has been insulated by insulating material of tungsten or silver chloride and some of the metals whose insulation could be carried out, and from that it has been taken as to be the potential A, and the reference electrode which has been placed outside the cell, from that it will be taken as to be the B. So the difference between these two potentials gives us the metal electrode biopotential. And so this is an electronic circuit which is equivalent circuit for the metal electrode, and this is a simplified equivalent circuit for the metallic microelectrode, and this is a real-time application for the metal electrode where it has a sharp or a very small diameter which could be inserted inside the single cell, and it has been insulated by tungsten in order to provide the any electric shock which will be affected for the patient. The second type is of non-metallic or the micro pipette electrode. It is also used to penetrate within a single cell. So it consists of a glass or a micro pipette which could be of a very small diameter of 1 micrometer. It has been filled with the electrolyte solution, and the stem it has a very thin wire which is made up of a tungsten or a stainless steel or silver, which that thin wire will be inserted into the cell, and the one end of the pipette will be attached to the potential which is taken as to be the reference A, and the reference electrode will be taken as the outside of the cell which will be taken as to be the R. The difference between these two potentials gives us the non-metallic or micropipette biopotential. This is the electronic circuit or the equivalent circuit for micropipette, and this is how the real-time micropipette looks. This is a single penetrated wire. This is a glass filament filled with a mercury, and this is a small wire, and this small wire will be inserted into a single cell, and outside the electrode which will have a reference electrode, the potential between these two gives us the micropipette or non-metallic electrodes. These are some of the applications of biopotential electrodes, where cardiac monitoring it will be used with silver chloride electrodes; sleep encephalography EEG, where it will be used with gold or silver chloride electrodes; for eye movement, silver chloride with hydrogel. For most of the biopotential electrodes, silver chloride electrode will be used as the reference, which is which has a very insulating capability and prevents the patients from electric shock. Thank you.